So with that, I'll turn it over to Jenny, who will give her presentation on kinase inhibitor mechanisms.
Hello. So I'd like to point out that I'm now at Takeda, California, but all of the work I'm presenting here is from Hoffman La Roche. So when I say we, I'm referencing the work from Roche and not from Takeda. So cardiac adverse events is a primary cause of preclinical drug attrition. Is there a pointer?
Yep, right here.
Okay. And this is in part due to inadequate animal models used during the drug development process. Therefore, there is a clear need for a better predictive model of not only cardiotoxicity, but also in general cardiac adverse events, such as arrhythmias. So current animal models that have been used, cardiac telemetry models, ex vivo models, such as the Langendorf, as well as in vitro models. And a better predictive model will not only allow for a decrease in animal usage, but also a decrease in preclinical attrition rates. Recent advancements in stem cell technologies have allowed for the differentiation of iPS-derived cell lines, including the cardiomyocytes. This novel model not only expresses functional contractile proteins such as troponin T, alpha-actinin, and alpha-beta myosin heavy chain, but also functional channels such as sodium, calcium, and although at low levels, the hERG channel as well. We also did a considerable amount of expression profiling of these cells, and we found that they were much closer to the fetal, which is in red, than to the adult cardiomyocyte. But if you keep these cells in culture over a long period of time, they tend to increase the expression of the adult cardiomyocyte markers. Therefore, we found that this model also not only has functional contractile properties, but also functional electrophysiological properties. This is a trace of the microelectrode array in which right here you see the sodium peak to the top of the calcium peak, which is the equivalent of QT. So the purpose of my studies, of what I'm going to be presenting, was to determine the mechanism of sunitinib, which I'm Sutent, mediated cardiotoxicity in this novel iPS-derived cardiomyocyte model. Sutent is a multi-targeted receptor tyrosine kinase inhibitor, which inhibits both the proliferative, survival, and angiogenesis pathways, and is currently in the clinic to treat advanced renal cell carcinoma and imatinib-resistant gastrointestinal stromal tumors, but is associated with a considerable amount of cardiac adverse events, such as hypertension, which leads to hypertrophy of the cardiomyocytes, as well as decreases in the left ventricular ejection fraction, as well as congestive heart failure. And also in the clinic, they've been able to detect prolongation of QT. The adverse cardiac events have been associated with the promiscuity of Sutent. It binds over 180 kinases, as you can see in the AMBIT profile here to the right of the dendrogram. This is a 10 micromolar AMBIT profile. So I wanted to take a look at a various number of kinases of which have been predicted in the literature, such as AMPK and RIS by Tom Force's group. And then looking at the AMBIT profile, which I'll discuss later, I looked at a number of kinases. The only one I'll discuss will be Aurora. So AMPK is the master metabolic regulator of the cell, and it regulates the anabolic-catabolic switch. Cardiomyocytes have a very small pool of ATP, and once it's depleted, the cell is most likely destined to die. So therefore, we thought it was critical to take a look at AMPK. But in the literature, there's been a lot of controversy over whether AMPK is mediating Sutent cardiotoxicity. So Tom Force's group, and this is a paper published by Kirkella, overexpressed a truncated form of AMPK in neonatal right ventricular myocytes and then treated with Sutent. And you can see that there's a clear attenuation of Sutent-mediated cell death as measured by the tunnel assay. On the other hand, Brian Hassanoff's group, they pre-treated with metformin, and then they treated with Sutent, and they were hoping that they would see an attenuation of Sutent mediated cytotoxicity because metformin overactivates AMPK, and he did not see any attenuation. Therefore, we wanted to move forward and try to test whether in a more relevant human model that has a more relevant energy homeostasis mechanism, whether Sutent is actually mediating the cardiotoxicity through AMPK. So first we just wanted to profile Sutent-mediated cardiotoxicity in the eye cells. So in an ATP depletion assay, over a concentration range of 4 to 250 micromolar and a time course from 0 to 48 hours, we wanted to determine a more narrow window to test the cells in further experiments. And as early as 4 hours at 31 micromolar, there was an 80% depletion of ATP, and by 24 hours, there was almost 0%, around 1% of the cells were still alive. So I was able to narrow the dose-response curve that I was going to look at in the experiments I'm going to discuss from around 8 micromolar to 31 micromolar. And I looked at a variety of different cytotoxicity endpoints, such as ATP depletion, right down here, glutathione oxidation, lactate dehydrogenase release, which is looking at the integrity of the cell membrane, as well as caspase 3,7 cleavage, looking at apoptosis, and found that the TC50 for ATP depletion in these cells was around 16 micromolar. So Sutent is inhibiting downstream signaling of AMPK and not the upstream phosphorylation by either LKB1 or AMPK kinase. So first I just wanted to confirm by Western blot that these cells are actually expressing AMPK and that we can in fact modulate its downstream signaling in this model. So right here is the expression of AMPK, and as a positive control I also looked in the HEC293 cells. Sutent is only supposed to inhibit the downstream signaling of AMPK. You can see clearly here that it's inhibiting phosphorylation of ACC and is not affecting the phosphorylation of AMPK. So in order to test whether AMPK activation can attenuate Sutent-mediated cardiotoxicity, I pre-treated the cells with potent activators, ICAR and metformin for 3 hours, and I just wanted to again confirm by Western blot that I can actually upregulate the phosphorylation of AMPK as well as its downstream target ACC over the time course that I was interested in. So I pre-treated with both 1 and 2 millimolar ICAR as well as metformin, and then treated a dose response of 0 to 31 micromolar of Sutent. And in an ATP depletion assay, I did not see any attenuation of Sutent cytotoxicity. So therefore, I concluded that AMPK may not be a critical kinase mediating Sutent cardiotoxicity, but that's not sufficient. So I went into the vast kinase library that Roche has, and I pulled out AMPK alpha 1 and 2 small molecule inhibitors that inhibit around 100%. And I treated from 4 micromolar to 250 micromolar, and I measured not only ATP depletion but lactate dehydrogenase, and there was no cytotoxicity in 2 of the molecules, and a TC50 of around 250 micromolar. So therefore, I concluded that AMPK is not a critical kinase involved in Sutent-mediated cardiotoxicity. The other kinase that Tom Force hypothesized may mediate Sutent cytotoxicity is RISK. So Ribosomal S6 Kinase was proposed because it had KD values within 0.14 and 0.017 for RISK-1-2, which was within the 0.1 micromolar effective plasma concentration of Sutent. And it also had, in a kinase assay, an IC50 value of around 0.36 micromolar, and this was work done by David, by Hassanoff's group. And it's also a target as an anti-cancer agent. It's overexpressed in 50% of breast cancers. So it would be good to know if you inhibit RISK, are you going to have any sort of cardiac adverse events in the clinic? So I collaborated with Jack Taunton at UCSF, and I got an irreversible potent inhibitor of RSK. And in an AMBIT profile screen, it's extremely selective to just RSK, and it doesn't have any off-target effects. So I did a similar time dose response from 4 to 250 micromolar, and I also measured similar endpoints of ATP depletion and lactate dehydrogenase release, and it had a TC50 value of around 250 micromolar. So RSK is not a cytotoxic agent in cardiomyocytes. But I wanted to determine whether it had any effects on the electrophysiology. So I utilized the Excelligence RTCA cardio system. So this is a measurement of impedance. It's a shift in the XYZ plane, and the trace that you see here is the functional contraction of the cells. At the top of the peak, it's fully contracted. So these cells are plated in a monolayer, and they're very dense, and they beat synchronously. And so the top of the peak that you're seeing here is when they're fully contracted, and they're lifted off of the cells, off of the plate. And at the bottom of the peak, they're fully relaxed. So what you're looking at here is a number of different wells on the Excelligence system. The top 2 wells is no treatment, and then vehicle, DMSO. And then below here, 3 wells where I treated at a low concentration of 12.5 micromolar of RSK. And within 5 minutes, I had a significant decrease in amplitude as well as decrease in contractility of the cells. So I thought that was kind of odd, and I wanted to look at it throughout a longer time course. And by 45 hours, I didn't see any rescue of contractility of these cells. But I knew based on the LDH assay and ATP assay that these cells are happy and alive. So I hypothesized that RSK may have an effect on the electrophysiology and contractility of these cells just based on this experiment. I wanted to take a look at it further and use microelectrode array, as well as look at similar effects of the electrophysiology using automated patch clamp. We're looking at inhibition of the sodium channel and hERG channels as well. But first I'm going to discuss profiling of Sutent. So again, this is a microelectrode array, and the top of the sodium peak, and here's the calcium peak to the top, is about right here, and that's the equivalent of QT. And this is a 10-second trace that you're looking at, and these cells are beating synchronously within the microelectrode array. And as around 10 micromolar, we see the initiation of arrhythmic beats, and by 30 micromolar, there's a decrease in the electrophysiology altogether. There's decreases in the sodium peak, the calcium peak, decreases in the beat rate, as well as the field potential duration, which is a measurement of QT normalized to beat rate. So again, from the sodium peak to the top of the calcium peak. I wanted to see what type of concordance we had between looking at the iCell model using the microelectrode array and well-established models that Roche has with CHO cells that overexpress hERG, as well as Chinese hamster lung cells overexpressing the NAV 1.5. And this assay was done on the Patch Express. So first we attempted to clamp the eye cells, but we weren't successful. So this is why I looked at this is why I utilized the CHO cells. So the IC50 of hERG was around 1.4 micromolar, and of NAV 1.5 was around 7 micromolar. So it correlated really well to what we saw in the eye cells in terms of the initiation of the rhythmic beats. So inhibition of the sodium channel correlated to the QT prolongation that we were seeing. On to RSK. With as low as 0.3 micromolar, we saw decreases in the beat rate as well as in the sodium channel, and by 3 micromolar, it had more significant decreases, as well as a decrease in the calcium as well. So similarly, I looked at using the Patch Express with the cells that are overexpressing hERG and the sodium channel, that I saw very potent inhibition of hERG, but a much less significant or not significant at all inhibition of the sodium channel. Therefore, it appears as though that the eye cells may be more predictive of the inhibition of the sodium channel than overexpressing NAV 1.5 in the Chinese hamster lung cells. Lastly, I'm going to discuss Aurora. So I compared a number of kinases that are critical to the heart, which is the list that you see here, to the AMBIT profile of Sutent. And any kinase that was inhibited greater than 85% coupled to all of the AMPK inhibitors, so I looked at their AMBIT profiles as well. So anything that was inhibited in the AMPK that was not cardiotoxic and did not have any cytotoxicity or adverse cardiac events, I ruled out. And so that's what you're seeing here highlighted in pink. And of the two, there's the Aurora B and Aurora C. Aurora C isn't expressed in the heart, so my focus was on Aurora B. So Aurora is involved in mitotic spindle formation, centrosome function, as well as cytokinesis. But in the literature, it also suggests that Aurora can phosphorylate AKT, which can modulate myocardial contractility as well as hypertrophy. So by Western blot, I just wanted to confirm that this signaling might actually be occurring. In the Sutent AMBIT profile, it was showing that Sutent was not directly inhibiting AKT, so we were hoping that it was through the downstream signaling. So here you can see that it's in lane 1 here is the vehicle, then the AMPK inhibitor, and the third lane is Sutent, and the next 2 lanes are the 2 Aurora inhibitors, and then a positive control is just the HEC293 cells. And you can see clearly that there is an inhibition of phospho-AKT, so then I moved on. So I selected a number of Aurora inhibitors. So these are pan-Aurora inhibitors. They were inhibiting to various degrees all A, B, and C, although C is not relevant. And there was some level of cytotoxicity in terms of ATP depletion and LDH release, but we've done a significant amount of testing of a variety of TKIs, and we found that unless the TC50 values were less than 20 micromolar, anything above that was not significant. So we didn't feel that this level of cytotoxicity was relevant. But I went on to look at Aurora B inhibitors. So this was very selective to Aurora B, and there was less than 85% inhibition of the other 2 Auroras. And there was no cytotoxicity, even up to 250 micromolar. Therefore, I concluded that Aurora B is not involved in Sutent-mediated cardiotoxicity. So the conclusions that we had were that these human stem cell-derived cardiomyocytes, that they're quite novel because you can look at, in a single cell, the cardiotoxicity or cytotoxicity of the cell, preferably using ATP depletion, but more significantly, the electrophysiological effects of a variety of kinase inhibitors. So this would be very relevant in early screening of molecules to try to rank order the compounds before you go in vivo. And that the Sutent profile, we were able to profile Sutent adverse cardiac events in this novel iPS-induced differentiated cardiomyocyte model, and that we did see a dose-dependent cardiotoxicity of Sutent and with ATP depletion of a TC50 around 16 micromolar, and that the electrophysiological and contractile effects we saw as low as 1 micromolar, and that we also saw a relevant reduction in the sodium and calcium and beat rate, as well as prolongation of the QT interval using the microelectrode array. And that although it inhibited AMPK signaling, it is not critical in mediating Sutent cardiotoxicity. And also Aurora is not a relevant mediator as well. So although we saw a potent sodium channel inhibition in the iCells, we did not see it in the Chinese hamster lung cells. And it was also further confounded, the interpretation of the MEA, because of this potent hERG inhibition. And lastly, Sutent cardiotoxicity, it may not be attributed to the off-target inhibition of a single kinase, but it's more than likely a synergistic effect compounded by the inhibition of many kinases, because it's not very selective. It inhibits over 180 kinases. And with that, I'd like to thank everyone at Roche, especially the director of the Early Investigative Safety Group, Kyle Colliget, as well as Jack Taunton for collaborating with me on using his irreversible risk inhibitor.